It's a bit late for that.
Yeah, but yeah, the first half was reviewing.
So looking back, we should have constructed also the Benchling schema. We should have added it to the agrobacterium transformation soy and maize, and then have a metadata field for early sampling or so we should have done that. But the reason why we have it in separate schemas is because in science, those are completely separate teams. And they didn't. So when we brought it to one schema, then they want to have a unique code for their experiment names. And if we brought it together, they couldn't keep their way of naming things. So I agree that you can bring it to the same schema, as long as it's in the database clear that it is early sampling, because the sampling moment is extremely important for the data analysis, because the earlier you go, the more mosaicism you have, and you need to know that for analyzing the data.
Sorry, can you repeat that? The earlier you go?
So the plant grows, so the thing goes from an explant completely to a plant. So when you have the plant track, you sample when you have a full-blown plant. When you do transient, it's somewhere in between. But what happens, it's on selection, which means that the number of cells that are untransformed will decrease. So it's important for the AMP-seq result, it tells you how many reads have edited or edited. So you need to know at which moment in time it is being sampled, because you expect an categorical rather like bins when you have a plant, but earlier in the process it's like a smear, the results. So you need to know. Yes.
Interesting. I didn't know that. That's cool.
Yeah.
Okay. Yeah. It's here. Okay, so we decided we are going to include all of these three entities in MRD as a plant table, like this transient plant and external plant. We'll add a new table for the plots. Okay, let's go forward with this. Protoblast will be its own entity, right? Because it's a different concept. It
completely different. No.
Okay. Yeah. Okay. So protoblast, you mentioned like it's a plant-like.
It's a cell, a plant cell, where you bring in all the editing machinery and then you do a readout even in the same week. So it's a very quick system to test your guides, for instance. So you isolate cells from a plant, you transfect them, so you shoot in the editing machinery, and then after a few days you take the DNA and you check how many of the reads are edited via AMP-seq. That's a quick testing system.
Okay, isn't this in LabVantage equivalent to what they call it DNA sample?
No.
It's not equivalent. Okay.
So is this the one where they, like remove the cell wall and then
Yes.
do the transfection thing? Okay.
Yes. That is this. And that is what product is requesting from science when they want to, for their transformation, you test your guides for their efficiency in a quick system first. And that is what we do for them.
Yeah. So basically this cannot become a plant even if you let it grow.
No.
Okay. Yep.
Okay. So yeah. So this will be on its own, right?
Yes.
Okay. And okay, so we don't have any parents for this generic biological sample, right?
That is an entry point for us. So if they send over things from West Lafayette that are not registered in our system, but that we want to analyze with our pipeline, we need to have fields like nuclease guide. And that's how we register things that enter our science organization, but without being registered in Benchling beforehand, which sometimes happens.
Okay.
Okay. So okay, let's take a look on this protoblast. So what is the important information we need to capture here?
You have the protoplast batch. So that's basically an identifier for a bunch of cells that you start with, and associated to that, so that protoplast batch, you will see if you go to the protoplast batch entity, is linked to a plant, and that again is linked to a seed, and you take the cultivar from there. So the important information that you need to have is the cultivar. It tells you which genotype the experiment was in. So you will see if you go there, protoplast batch, that is an entity in Benchling, and that is extracted from a plant. So if you go to this entity, you will see one of the fields will be plant. And if you then go to the plant, you will see that there is a cultivar associated with the plant. So in reality, how this works is you grow plants and then you extract the protoplasts. You sow plants from seed and then you extract protoplasts. So then they create a protoplast batch. So that determines the cultivar in which the results are being generated. And then the other relevant information are 3 metadata fields: the Cas protein batch, the guide, and the plasmid. That are also metadata fields on this. Yeah. So you see there, Cas protein batch, plasmids, and cRNA. Yeah. Go ahead, Kishore.
Do we need to track the plant as well here, or just the cultivar? What do you think?
I think the cultivar is okay.
Okay.
Yeah. Then I will look. There are okay. Sorry.
Yeah, I have a question. You said like that's a protoplast experiment condition, and then here we have this protoplast batch. Protoplast batch refers to this protoplast batch table,
Yes.
which has where is the plant? Plant use. Oh.
Yes. No, that's the number of plants used.
Yeah.
Plant ID number, protoplast batch. That almost looks like he's looking at a result table.
Yes.
Protoplast batch. Plant pot. Yeah, I don't.
There's a result schema. We can check in our. You have Benchling open, Audrey. Can you check the protoplast batch? Protoplast batch, but there is a plant. Plant ID.
Oh, plant pot. There we go. Yeah, you want the plant pot field. That'll link you out to the plant.
The warehouse name is a bit
confusing.
And this plant information or this plant isn't in our Benchling, right? Like in any of these plant tables.
Yeah, that's in the plant table. Yes, and you go to the plant table, and there you will see on the plant table you have a cultivar field. So you take just the cultivar field on the plant.
Okay.
So plant pot links to the plant. That's a plant ID that you will find there, and then you just take that plant and you take the cultivar field, and then you have the cultivar.
Okay. So yeah, I asked this question because we need to understand the relation. So now protoplast experiment condition will extract this metadata and it will have a reference to plant table. Okay.
Yes.
Okay.
So the protoplast batch ID and the plant ID is irrelevant for the end user. We just need to have the cultivar because, I wouldn't. It's irrelevant. And then on the protoplast experimental condition, so that's your plant material for your assay, and then you have the editing. You have 3 types of editing. Yes, you see there, Cas protein batch, guide, and then plasmid batch. So I will explain to you so that you understand it. There are 3 ways of doing this kind of experiment. So 3 different ways. So you can take your protoplast and you can transfect it. So you can add a plasmid only. That's the first way. So then all the editing machinery is on the plasmid. The second way that you can do the experiment is that you take your cells and then you apply a Cas protein batch and a guide. That's what we call RMP, that combination. And the third one is if you combine a plasmid batch with a cRNA, and then you call it hybrid delivery.
Mhm.
So depending on the combinations that are being filled out, you know which kind of protoplast experiment this is. So either plasmid only, or a combination of Cas and cRRNA, or a combination of plasmid and cRRNA.
Okay. Okay. Yeah.
You will see that, yes, that's relevant information. If possible, we don't have it as a metadata field, but if you would be able to code that, so write a little bit of logic, so that's the protoplast transfection method. It's hybrid, plasmid, or RMP.
Okay. So yeah, let
Yes.
me rephrase this.
Yes.
So now this protoplast experiment condition will have this metadata like protoplast batch, plasmid batch, Cas protein batch, and cRNA. Okay.
Yes.
And we need to add something like an indicator field or a new field
Yes.
that indicates what kind of treatment or what kind of
Yes.
experiment we have done, right?
Yes.
And the logic for this is, can you repeat this because I missed it?
Yes. Can you go to your sample data? Then I will explain you, because there you will
Okay.
see. Yeah.
Let me share my screen.
Yes. Okay.
Sample data. So we need
If you
to
go
go.
to the protoplast experimental condition, yes, and there
Okay.
you go to the sample data, yeah. So
Mhm.
you will see there, you have to the right, you have the cRNA Cas and voila,
Mhm.
RNA
Yeah.
Cas. So you will see, so if only plasmid batch, the first one is being filled out,
Mhm.
and the other lists are empty, now we call it plasmid transfection.
Okay. This one is plasmid transfection, right?
Yes.
Okay.
Then, if the first and the third one, so plasmid and cRRNA, are being filled out, we call it hybrid.
Okay.
If the second and the third one are being filled out, there will be no, because they made a mistake. There will be no, they have to update. I noticed that they made mistakes. So if the second and the third one are being filled out, we call it RMP.
RMP. Okay.
Yeah, that is empty for now, but I'm working with them to update. So in some cases, they filled out the plasmid instead of the Cas protein batch, so that will be missing. But as of next week, hopefully, you will see RMP experiments as well.
Okay. And what about this one? Nothing filled.
That is a control. You see to the right, it's a mock. That means that it's an untransfected. So that is for our internal process control. That doesn't need to end up necessarily in the database, because that is a control.
Okay.
You also will not have, probably, you will not have for this row, you will not have AMP-seq results.
Oh, okay. Okay.
Yeah.
Okay. So yeah, I think that's clear. I think yeah, to wrap up, will include the name. Nothing of this information is valuable, right? PSR,
The
nothing.
experiment date can be handy.
Experiment date. Should we include this mock as indicator
No.
or no?
Can you
Should we
leave?
filter on top of this one? Because you said like mock
So,
is something
yeah.
we shouldn't include,
Yeah.
right?
So what we are right now doing is we are going to change this Boolean column to a one with more different values that will indicate whether or not it's a negative, positive transfection control. So I wouldn't do it for now, and then later on we can still decide. This will be condition type. Can be relevant for your database, but if possible, can we wait a bit until we settled in science on what the value should be?
Okay. Okay. Yeah, we'll include this protoplast, plasmid patch, Cas protein, and cRNA. All of these are references, right? So
Yes.
this protoplast patch will refer to this table, right?
Yes. So for the
Uh,
protoplast batch, you only need to extract the cultivar. Eh? So the protoplast batch ID shouldn't end up there. Only the cultivar is okay.
Okay, so we'll get the cultivar from protoplast patch.
Yes,
Plasmid batch.
that's a pin number.
Yeah, this one.
Yes, yeah, that is a plasmid batch, and for this one, you have to take the parent plasmid. So if you go to a bit down, so the plasmid batch will be a physical representation of a plasmid, so something that we have in the lab. But you need to extract, so you go from the plasmid batch, you take the parent plasmid, and that will be a pin number.
Okay, so parent.
Yeah.
It's this one.
Yes.
Okay, so this will refer to the plasmid table.
Yes, that will be a pin number.
Okay.
Yes.
Okay. And let's go back in here. Yeah, so plasmid Cas protein. Yeah, I don't see it. So
Yeah, that is still, yeah, that I need to work. I'm working with the team. They made a mistake. So there should be yeah, that's a bit more complex. There should be a Cas protein batch. Can you go to the Cas? So I will, this is, we need to hop through a few schemas, but Audrey, wouldn't it be handier, like, this is the Cas protein batch that we just listed, and then the information that we need to pull from this is the nuclease. And that information will be in the child of this protoplast experimental condition. So we can take it from there. Then you do not have to compute yourself, Albert. So from this schema, we just take the Cas protein batch, and then from the protoplast sample, I will show you there is information that you can extract from this Cas protein batch, but it will be in the child.
okay, so you're saying like this information, like this will be the real information will be in the samples, right?
Yes, the
will be in
information
here.
that will be in the protoplast sample, because there in Benchling, can you go to the protoplast sample in Benchling? We will show you. So the Cas protein batch will encode a nuclease, and that nuclease will be listed in, yes, that nuclease will be a computed field in this one.
I'm gonna
So
go
if
all the
you
way to
look-
the end.
Okay.
Yeah.
Nucleus.
So yes. So maybe Audrey, I'm now thinking, so what
Yeah.
you can do, yeah. So you see all the information, like the cultivar is already here. So the computed field is exactly what we told you. So it goes from the batch to the plant to the seed, and it takes the cultivar there. So you can take the cultivar. Maybe that's an easier solution.
Should we move this information to the samples? Or
Well, can you combine the two schemas in one, Albert? So now in Benchling, it's two separate schemas, but maybe that is not needed. Like in your database, you can just have a protoplast sample, and then there is some information on the parent, on the condition, like the type of transfection that I explained you, where you have to combine certain metadata fields to understand. Can you calculate that over there, and then you just can take the other information from this protoplast sample.
Okay. So I think I prefer to do these separations. Yeah.
Okay.
Yeah, but yeah, I think as you mentioned, like there is some information in the samples.
Yes.
The question is, should we replicate this information, like put it in samples and protoplast experiment condition, or should we keep it in one of them?
I would keep the cultivar in the sample, because you do something similar for the plant sample as well.
Okay, so
you have it in plant.
yeah, but I think we were talking about this
Yeah.
the protoplast.
Yeah.
We were talking about these
Yeah,
two.
the condition. Yes.
Yeah.
So then you just add the Cas protein batch.
And this will be referring to the sample, you said? Or referring to what?
The Cas protein, what do you mean by referring to the sample?
So for example, we said like this plasmid batch will be referred to plasmid batch table, right?
Yes. Cas protein batch will refer to a Cas protein. That's also a schema.
Yeah, I don't see it. So here, Cas.
Let me check. It
Yeah.
will be probably nuclease.
It is nuclease underscore batch.
Okay, this one.
Yes.
Okay. So yeah. Okay. So this will be referring to nuclease patch, CRNA, CR patch as well, right?
I think that's the CRRNA itself. No, I should double check it.
For the
One of both.
experimental condition, it goes to the guide directly.
So the CRRNA then, Audrey.
Yeah, it
Yes.
goes directly to the guide. It doesn't go to like a batch or anything. It just
Yeah.
goes directly to the guide schema. Or
Oh.
CRRNA. It's
CRRNA.
CRRNA. Yeah.
Okay.
Just for-
CRNA.
Guide is easier to say.
Yeah. We have guides, so that's yeah. Okay, so this CRNA goes to CRNA, and okay, Cas will go to nucleus patch, plasmid will go plasmid patch, protoplast will go to protoplast patch. Okay, and we'll include conditions. We include a new column. Let's call it what? Like
Condition
the,
description. You want to copy it to the database?
no, I mean, do you want it or not?
Yeah. Do you? I think it's yes, it's needed because it sometimes discriminates rows. Otherwise, you will not have unique rows. So when they test, when all the other columns are the same, but they test, for instance, a different way to seal their plate, then you need to have the condition description to discriminate. So yes.
Okay. Yeah, so we'll include this. We will create a new column, the one we mentioned, like
Yeah, for plasmid hybrid or RNP.
so what should we call it?
I think it's protoplast transfection method. So transfection method.
Okay, transfection method. Okay. Okay, so now we finish this new entity. Yeah, now we still have all of these 4 plant samples or types of plant samples. Should we include all of them in one table?
I would keep so you can combine transient plant sample and plant sample in one, but protoplast needs to be separate, I think, and generic biological sample as well, because they link to different parents.
Okay. Okay.
Or
In
not.
MRD
Yeah, it could be. This one doesn't have parents, so like it's an orphan entity, right? But these 3 like could be included in one table if they have the same schema. I'm talking now about the schema of the concept of plant sample, like
Yes.
because when I looked at these tables here, like this is a schema. It's almost seem the same, right?
Well, on a plant sample, you have fields like developmental stage, tissue, sampling. Yeah. You see?
Yeah.
Yeah, so that is very different from a protoplast, whereas you only have the condition and then I think the sampling data. So it's super simple for a protoplast and you have completely different metadata fields.
Yeah. So protoplast is this one.
Yeah.
We have harvest date.
And the condition, that's it.
Mhm.
The rest are computed fields. But you see for the plant, the second one, it is
Yeah.
like an tissue developmental stage, tissue type, plant developmental stage, sampling. Yeah, the plate is the same.
Yeah, so let's take a look on what we have in MRD. Okay. So plant sample table, let's open the database. It's moving. This one's- Yeah, we have this sample ID and plant ID. So plant ID is the parent in our situation, right? It's a plant. And EXP is immature plant or
Plant,
X
yes,
plant. Yeah.
plant. Yes, yes. Okay.
This, yeah, this sample ID would be the sample name. I think in our situation would be
Biological sample.
like the file register ID, right?
Yes.
Yeah, so the question, as you see, like in plant sample, we refer to both immature and mature plant. I don't know if, but because it's a sample, yeah.
But then, like plant ID, like in some case, if you would have like to have the protoplast there, then the protoplast sample identifier should be the second column, and then in theory, in the plant ID column, you should have the plant experimental condition.
Conditions.
Yeah, so then like the plant is at the same level as the protoplast experiment, eh?
Yeah, but we have this column called sample type. In this situation, we can add like protoplast or
Yeah. Yeah, but
any.
let's say, yeah, yeah. So let's say that you have a protoplast, and in the sample ID you fill out the protoplast identifier. Eh?
Exactly, yeah.
But then, yes, but then in the plant ID column, then you
Go
should
to-
fill out there the protoplast experimental condition.
Yeah.
So
yeah,
then
yeah.
you should rename maybe the column or add an additional column to fill out, because that's
yeah.
not a plant, eh? A protoplast experimental condition. You see, in order to be able to fill the parent out in the first column, that is a bit
Yeah. I got your point. Yeah.
Yeah.
So the name of the column, yeah, it's not a plant. So it's okay.
no, and it's also not a plant sample. So
Yeah,
it's
a
very
sample ID.
confusing. Yes. So either you change, like you change it to biological sample, and then this becomes plant or condition ID, the first one, and then you can host everything, or you have a separate schema for protoplasts.
Yeah, I think there is a let's look at it in a different way, because as you see in MRD, we have different sort of information like tiers of A tier, MSIG, all of these tiers, and transgene status. Is it? I don't see any relation between this data and the data you have, right? Like for example, here, let's take this plant sample table or plant sample scheme.
But there will be on the plant sample, there can be information. I think there can be additional information. Eh?
Yeah, I
Like
see,
if you
yeah.
take a plant, whether it's a leaf or a root, then where is this in MRD, or is this information not being copied in MRD?
No, we don't have it right now. On a sample,
Okay.
yeah, all of this phenotypic information, you will not find it
So-
in MRD. But yeah, that's the question.
Yes. So either because then your table in our case, because we don't have the other information, then it's just making the link between a
Mhm.
plant and its sample. Eh?
Yeah, so this table only like make the link between the plants and this plant sample information. I don't know this stuff with other
Mm.
essays like MSIG,
Yeah.
all of these other essays. That's the
And-
job of this
Yeah.
table. But
Can this
if you want,
become? Yeah.
yeah, but what
Can
if
this-
you want to capture something like more information? I think we should, yeah, modify this.
Yeah, or you make it biological sample, then you have your sample identifier, that's a sample ID. There you can either have any of our, and then you have columns for the origin. Can be a plant, can be a protoplast experimental condition, can be empty in case it's a generic one. And then product just fills out the plant and the sample ID. And if it's another type for us, then you fill out the sample ID and any other column.
Okay.
Then you can keep your schema, and you just change it to biological samples.
Yeah. So
Because
yeah,
what is-
let's return back to this. So do we have this information in Benchling, like tiers? I don't think so, right? Like
what
fragment
is
and-
it?
This is a fragment analysis tier, MSIG tier.
But what is an MSIG tier?
That's a good question.
What is this information?
I don't have an answer for this question.
I think it's like their editing efficiency, how they can tell what plants took what edits, or if the plant took edits. Like a low
That-
LOQ stands for low quality indels. High quality is HIQ. I'm not sure the NSEs. Maybe like non-selectable or something.
So that basically when you have the AMP-seq results, you analyze and then you label the plant and then you push it. So it's like a label
Yeah.
on the.
Yeah.
But shouldn't that? Yeah.
Yeah, that's why I'm asking this question. Does it make sense for you?
Well, so I would be hesitant. We are not there yet. So what we need to do is we are now reworking the AMP-seq pipeline so that as part of the pipeline, we can give allele information and we can do this kind of binning, but we don't have the code. So if we do it now, yeah, people will just fill out anything and I wouldn't do it. So we don't have the information.
I think, yeah, one more thing is very important is the transgene status. Also,
Oh.
that's something like if
Yeah.
it's positive or negative or failed because they can get it.
What is a transgene status, Albert?
Is a transgene like if we have like they select the transgene from the parents if they found it in the child plant, as I understand it. So
So
they
a
mark it as positive.
transgene. Yeah.
Yeah.
So a transgene. So I'm confused because we are doing editing and a transgene is, for instance, if tDNA is inserted or if you work with like material from outside that has already transgenes from
Yeah.
competitors. It's like that?
Yeah. If you got like something like a plant from external location and you do some editing and you found like this transgene, the main gene from the parent is still in the child. So
Yeah. That again, that's an outcome of a Tuckman analysis. So you have in your molecular results database, you have qPCR
Mhm.
assays, and those are being used to determine the transgene status.
yeah, this qPCR says
Yeah. And there is also qPCR recommendation that is going to, or that is already gone. Yes. So this is basically an assay to determine the transgene status.
Mhm.
So I'm very confused by having those two fields in the plant sample, because what I would think is that you would link them on the plant, or I'm very confused. What are they doing there?
I think the one on the plant sample is something like for as indicator, right? To indicate it's true or false. It's not something like I think if you want to keep that
Yes.
for example, you can find it here. Transgene is whether the plant sample carries transgene or not. Selectable marker and Cas INQ are considered.
Okay, INQs, that's the identifier of the qPCR that we were just looking
Mhm.
at. So
Yeah,
that is,
and
yeah.
how we calculate it.
Yeah, so you look at the qPCR results. So I wouldn't hard code it. What I would do is just you can make a view on at the plant level where you collect the qPCR results, and then you can make it visible on the plant because this can change. Sometimes assays are being rerun, and then in theory you should update your plant sample. You know what I mean, Albert? So
Yeah.
this should be a view or something that you compute. And also that is hard coded. I'm not very sure, like those thresholds, they change from time to time. So
Yeah,
I
but
think this is
so yeah, what you're saying is this transgene status will be on the plant level, right?
yes, because it's not
But
at the plant sample.
but in theory, as you mentioned, every time you take a new sample or you do a new assay, you create a new sample for this, right?
Yes, but I can. So the question is, when do you update? Because those assays are being run independently. And the issue in Gantt is that sometimes we do the AMP-seq first, and then we do the TACMAN later. So then we do not have the information available when we create the sample. You understand what I'm saying? It's a different information coming from a different assay. Having it in that table is a bit problematic. So it is plant level information on the genome. So it's way safer to combine the qPCR with the plant schema and then just code the logic and then to look up the because what they do I think their process is they do a qPCR analysis and then they do the AMP-seq. But in science, we do not follow the same process. So we do sometimes the AMP-seq and we do the TACMAN weeks or months later. So then we have issues.
Okay.
So it's a bit more complex. And also at the plant sample level, so I understand I understand why they want to have it to interpret the AMP-seq results. It's handy if you know whether or not the editing machinery is in there. So if you have the status, then you can already know if it's negative, then if you see editing, that's very strange because without editing machinery, you cannot have editing. But it causes like issues if you want to, for science, if you want to have it, that's very, very hard to accomplish. You understand the?
Yeah. I mean, there is a huge difference in the process or the workflow
Yes.
between
Yes.
science and product.
Yes.
And this setup fits like the product workflow, right?
This fits the product
but
workflow.
but
And
it
also
doesn't fit you.
no, and also I do not agree that it's information that should be stored at the plant sample level. There are many arguments against that, I think.
Okay, so that's all of this related to the transgene status, right?
Yes.
You say like we can include this information only in the plant level,
Yes.
not the plant sample. And plant level, does it include all of these entities like transient and protoblast as well, or only this plant and external plants?
It's only relevant for the plant, the external plant, and the field trial plot.
Okay. Okay. Yeah, this will be on its own. So it's a different
For
entity.
the others, it's relevant, but not for the protoplast.
Okay. And how we can get this information in the plant? Where can I find it?
Yeah. So on the plant, there will be TACMAN results published. So in your schema, it's qPCR. And in our, I think it's copy number, it's a result schema. And I think copy number or TACMAN results.
It is TACMAN copy number PCR V1, I believe.
Yes.
TACMAN?
Yeah. So you go to the
Q.
with the Q. Yeah. So you take a plant. Yeah. qPCR version. So if you go to the sample data, so what you will see there is that the first field, the first real field is the plant or a plant sample. Voilà.
Yeah,
This
Biological
biological
sample. Yes, will link to a plant or any other biological sample. And then you have the result of the TACMAN. So the number is in the result field. So we have three different, in one row, you can register results on three different genes, on three different elements of the transgene. So do you remember the threshold? So they said if it's a value above 0.5.
Yeah, it is calculated on top of copy ratio.
Yes. So you
let
can
me
just,
open it here.
yeah, you can put logic there.
Yeah, so I think now we are talking about this qPCR table.
Yes.
qPCR
And beforehand,
says,
yes. What you did beforehand was, yes, what you did beforehand, you had a qPCR recommendation in there.
we just remove it. We removed it because
Yeah.
we had a separate recommendation table for this.
Yes.
And yeah, they said like it's not useful for them.
Yes. So the
So
qPCR is similar to that.
Should we? So that's leads us to another question. Should we include data from
Yes.
TACMAN copy number PCR
Yes.
to our qPCR assay table?
Yes. So what you have, yes, but that is not AMP-seq anymore. So that's a different.
Yeah, it's a qPCR. Yeah, because we have
Yes.
qPCR and AMP-seq. It's
Yeah. So what we are doing now is that you have qPCR assays, and the assays will have a unique ID. They already have an INQ number in product. And then what we are doing in the coming weeks and months is we will use Benchling as the system of record for those assays. So those INQ numbers will be registered in Benchling in their separate schema, and we will link them to the Stackman copy number schema.
Okay.
So we will publish, yes, that's what we will publish in the coming months, version 2, with a link, a unique link to the assay. So I would hold, or I would wait until we have that implemented, because that will make your life a lot easier.
Yeah. Okay. Okay. Yeah, I like this. So yeah, I think we are over time, but still we haven't talked about this plant sample. So that's what I'm going to do. I will take a deeper look about this different plant sample, all of the information we talked about in this meeting. Try to understand all of these relations, and I will send an invitation for a different meeting to continue talking about plant sample. Is it good or
Yep.
okay? Yeah, that was very useful. I think we, for the part of plants and protoplasts and the other conditions, I think we are good.
Yeah.
Yeah, just
Yes.
one step to MSeq. I think MSeq will be pretty straightforward because it's an
Yes.
essay. We'll
It's
include
safe.
it to our essay. Yeah,
Yes.
we have this plant sample stuff to iron out, and yeah, we're ready to go. Okay,
Okay.
thank you.
Bye bye.
Thank you, team. Bye.